So it's a great pleasure to be here in my beloved India. You all are so talented and you accomplish so much, and the future really is in front of you. I was touched to read in the opening book that Daniel Mazia is mentioned quite a lot. He was my mentor, and I like this picture of Daniel because he's holding two sea urchins, which you can think of as the spindle poles. He's maybe the chromosomes. And he was prescient both in terms of his views on the centrioles, but also on his hairstyle. You can see that's rather popular even nowadays. And Daniel had a great historical perspective, and he would talk about how Lamarck a few centuries ago would ask the question, how do beings of a complicated structure such as spontaneous generation could never derive their existence? And for Lamarck, nothing was easier to be conceived. He wrote that if the orgasm excited by this organizing seminal vapor is prolonged, it will augment the consistency of the containing parts. And this will render them susceptible of reacting on a moving fluid which they contain, and an irritability will be produced, which will subsequently be possessed with feelings. The first efforts of a being thus beginning to develop itself must tend to procure it the means of subsistence and to form itself a nutritive organ, hence the existence of an alimentary canal. Now you can appreciate that the great biologist Lamarck was French because he went straight from orgasm to alimentary canal, straight from sex to food. But the organizing seminal vapor, which he wasn't aware of, is what we will hear about in much of the rest of the session today, and that is the centrosome. And the problem of the centrosome during fertilization is a tricky one because in somatic cells, the chromosomes duplicate, the cytoplasm doubles, and the centrosomes double. And so you effectively have 3 cycles all going in unison. In contrast, in fertilization, the chromosomes during meiosis split to haploids and then recombine at fertilization. The egg gets virtually all the cytoplasm, and we think that the sperm brings in the centrosome, but the exact nature of that isn't clear. And so by studying humans, we could show that here is a human fertilized egg where you can see the sperm tail in the egg, and tails always come into the egg, regardless of what they say in your textbooks. And you can see in this human egg, there are 2 dots of gamma tubulin, just as you might expect. Later, looking at microtubules, you can see this is the sperm nucleus. There's an aster here, the sperm aster. Here's the egg nucleus. And later, the sperm aster enlarges so that the male pronucleus, the sperm nucleus, moves towards the egg, center of the egg, and the microtubules contact the egg nucleus. Now, imagine, and I don't want to fall off, imagine that we're all inside of an egg. You're all mitochondria, and there are a lot of mitochondria in eggs. And let's say I'm a sperm. That may be easy for you to envision. And I could enter the back of the room, or I could enter the front, or the side, or the top, or the bottom. And my job as a tiny little sperm in this huge egg is to find the egg nucleus. But look at the volume here. How do I find the egg nucleus? And the answer comes from that brilliant sperm aster. Because porcupines don't really do this, but if porcupines did it, as I enter the cortex of the egg, my microtubules grow out and they push me towards the center of the egg. And simultaneously, the microtubules are shooting out like harpoons. And when they contact the egg nucleus migrates down the sperm aster to reach the male pronucleus, and we can see that, hopefully, in this next slide. There's the egg nucleus, the sperm entered there, and there the sperm and egg nuclei are coming together, and the egg is going to divide. So here's another example. Look, here's a sperm entering. Watch how the egg nucleus just goes zoop right across the egg to reach the center of the sperm aster. I would like to think of this sperm aster as really your lucky star, because if you didn't have one, you wouldn't be here. And here the egg nucleus, the zygote nucleus now breaks down, first division. Let me show you one other. Here's again the sperm entering, and you'll see the egg nucleus, zoop, goes right to the spermaster, and then later the spermaster duplicates to form the two poles of the mitotic spindle, and of course, first division occurs, second division is perpendicular to first, third is perpendicular to second. And again, coming back to humans, human fertilization is just like almost every other animal with the exception of rodents. So the two centrioles, or two centrosomes separate. At first mitosis, again, the sperm tail is associated with one of the two poles. And remember that the problem, the problem of polyspermy is an imbalance between the number of centrioles and the number of chromosomes. So for example, here in a human egg that is dispermic, you have two sperm, you have a triploid number of chromosomes, but you have 4 centrioles. And these will tend to divide from 1 into 4. So as in C. elegans, as in Drosophila, as in Xenopus. So what I'm going to explain to you next is that we can now appreciate that the sperm brings a centriole-like structure into the egg, including in humans. And there are some men whose partners, when they go through in vitro fertilization methods, seem to have their eggs perfectly fertilized, but development doesn't proceed. And the reason is that there are problems with the functioning of the sperm centrosome. So that, for example, there are cases like this where the sperm doesn't form an aster, but it's entered the egg. There are others where there's a truncated aster, still others where the aster is separate from the male pronucleus. And there are mutants in Drosophila and C. elegans and elsewhere. And this issue that I'm talking about is one that Boveri already 150 years ago recognized, because he wrote that the ripe egg possesses all the elements necessary for development, save an active division center. The sperm, on the other hand, possesses such a center, but lacks the protoplasm. And so the question now is, how does the sperm centrosome, centriole, and the egg components meet one another and form the zygote centrosome? Because you really need a zygote centrosome. And what happens is that the sperm basal body is severed from the tail. A lot of gametubulin is recruited. And now with the advances in super resolution microscopy, we can begin to ask questions about when does SAS6, when do many of the other molecules that we now recognize as critically important for forming the centriole, when do they come in? And working with Jadranka Larankic and Tomer Avor-Rees, we've been looking at human sperm during fertilization, and we can see that there's a structure right at the base of the sperm which has both centrioles embedded, though one is degenerated. And this structure right by the sperm head actually looks like a funnel or a basket, and it enlarges after the sperm enters the egg. And here we can see the sperm and egg nuclei together. You can see these splayed microtubules at the head of the sperm with centrin forming a perpendicular structure. It remains on one of those poles during first division. And even further, we can follow SEMP152 that was brought in by the sperm and begin to characterize the molecules that are being brought in by the sperm and which are being recruited by the egg. And in fact, SAS6 is now recruited within the egg to the SEMP152 template, and we believe that's giving the zygote the very first centrioles. And so just as you're going to hear about extraordinary studies in C. elegans and other systems, SAS6 is vital for forming this pinwheel, and we now are moving forward in understanding what's happening in human reproduction and why things fail. And I need to say that I love being in India because I have so many dear friends. T.C. Anand Kumar was the pioneer of the first scientifically documented test tube baby here in India. He passed away a number of years ago, but just before a little bit before he died, he wrote an article about Dr. Subras Mukherjee. Dr. Mukherjee made the first in vitro fertilization baby in India and perhaps succeeded with the first IVF in the world. And it's a tragic story of science. Unlike the accolades that Bob Edwards and Patrick Steptoe received, Mukherjee received nothing but humiliation from the West Bengal government and his colleagues. They didn't even allow him to publish his data and refused permission to travel abroad. After he claimed success in '78, the government set up an inquiry. They stopped his research and transferred him to an eye hospital. He was harassed from many quarters. He was denied leave to write his responses to the panel. 44 days after the transfer order, he committed suicide with a ceiling fan in his office, bringing an unceremonious end to what could have been among India's biggest accomplishments. It's really a tragedy when colleagues gang up on colleagues. And I'm glad that the history was written properly by T.C. Anand Kumar. Now, I'd like to stay with the sperm. And for the men in the room, and you know who you are, your epididymis is over 20 feet long. You often think of the sperm after it's coming out of the testis as having finished its work. But that poor little sperm, it now has to go through another 20 feet before it has a chance. And I mean, it's real, and it does it within about 10 days. And it's really a heroic effort. And recently we showed that centrioles in the epididymis are lost. First one, and then a second, as measured by centrin. And this is also where much of the imprinting RNA is added to the sperm. So don't think that the sperm is fully mature after it leaves the testis. There's a lot going on in the epididymis that we don't know anything about. And the amazing thing about studying human reproduction is that you're working with real people. And this is an individual who's transitioning to be a man, but before he had the elective surgery, he asked his gynecologist to help him have a child. The testosterone was dialed back, and here he has his little son. And in the transgender community, lactation is referred to as chest feeding, not breastfeeding. And to me, this speaks to the wonder of social media, that people who couldn't previously find someone to love now are able to. And for the doctors in the audience, remember that before this man has his surgery, he's going to need a pap smear. And in the reverse direction, a woman who hasn't had her surgery yet would need a prostate exam. And these are counterintuitive. Now, there's a much larger group of people who are men who suffer from genetic infertility. And, you know, we often think of the power of molecular genetics as preventing the propagation of mutations. But in the field of reproductive medicine, mutations are propagated, and there are all sorts of Y chromosome microdeletions and other methods that are used to propagate deletions. I like this slide on the genetics of male infertility. I don't know why the French used this structure, why they had to give it Viagra, but you can see it's circumcised, which I like. And so there's one technique known as intracytoplasmic sperm injection, where you inject a sperm right into the egg. And we made a lot of monkeys this way so that we could study what's happening during this unusual type of fertilization. And the way that the sperm enters the egg isn't that it's entering through the cell surface, but it's injected right into the center of the egg. So the acrosome remains on the sperm. Nuclear decondensation is all irregular. You can see some pores coming in. The X and Y chromosome are non-randomly positioned here. There are structures that are normally lost at the cell surface, but they aren't after ICSI, and they stay on in the egg. And let me just show you an animation here, where during ICSI, what happens is a single sperm is aspirated into a fine micropipette. We used to use the verb suck, but since Clinton left office, we use aspiration. Oh, you're a tough audience. Okay, so the egg is held by a micropipette, and that sperm is introduced to the center of the egg, and somehow development starts. And it's really quite remarkable. And furthermore, it's remarkable because there are modes of selection which play some role in determining which sperm will enter the egg, whereas with ICSI, someone looks down a microscope and goes, ooh, that's a cute one, and just nabs it, and that's the paternal origin. And there are probably 6 million babies on Earth made by this artificial method, and we know the choreography is different than natural fertilization, but we don't know what the long-term consequences are going to be. I want to switch to the female for a second, and I want to mention that we've recently found the egg centriole in unfertilized eggs. We can see it here by GFP Centrin, and you're seeing the last gasps of the egg centriole as it's going to be lost. And reproductive aging in women is a major issue. It's very different than in men. A Harvard professor in 1873 wrote a well-reasoned book on female infertility. The culprit, he said, was the education of women, which diverted energy from the reproductive machinery to the brain, resulting in women with monstrous brains and puny bodies. Now, this guy obviously didn't divert any energy to his brain. And, you know, you need to worry about what's going on. But there are a number of clinicians that are trying to turn back the clock on an egg from a woman who's maybe 45 or 50, and they think there are some magical substances in young eggs, and often they will say, look, if you take a flashlight and put it on a shelf for 40 years, the battery will run dry. What's the matter with you guys? All we're doing is taking mitochondria from a young and juicy egg and changing the battery. Now, I know a little more about mitochondria, and they're not just batteries. They do a lot of things. And yet, for $25,000, you can go to a number of places on Earth and have young mitochondria put into your egg. The pregnancy rates are nearly identical, but there are companies that are valued in the hundreds of millions of dollars by claiming they're going to put juicy mitochondria into old eggs. Now, there is a special circumstance where women who have a mutated mitochondrial DNA can get, in the UK, a healthy donor, and this you may have heard of as the 3-parent babies, where you have the nucleus coming from one woman, the cytoplasm another, and then the sperm coming from the presumptive father. There was a live birth resulting from this spindle transplant method. It was done in Guadalajara, Mexico, although now there are questions about whether it was done in New York, but the FDA will sort that out. What they did is only transfer male embryos. The notion being that since the sperm does not transmit mitochondria, the heteroplasmic mitochondria should only influence that one generation and not beyond that. But there are other groups that are doing other things. You've all heard last week about how you can clone monkeys. The results are not impressive, but we are walking down a slippery slope. And this is a slide from an infertility clinic, which is quite remarkable. They're suggesting that you can have a child with an IQ of 130 or have a child with an IQ of just 70, and they claim that for $25,000, they can raise the IQ of your child. I'll point out that the children end up as attorneys, chemists, and executives. No scientists, no teachers, forget about it. But this is nonsense, and yet it's being marketed already. And so we're in a weird world where infertility treatments are paid for by cash, and in the great means of capitalism, money talks, and you can do whatever you want, regardless of how crazy it is, and no one's really representing the child. And so it brings me to one of my last slides, which is here, and this reminds me that we all look through the lenses that we were raised with, our own faiths, our own traditions. These are sensitive issues, and I don't know that my bioethical view is necessarily the correct one or yours is incorrect. And I like this slide. I had problems in Saudi Arabia when I used this, but it's only people who have filthy minds that are unable to see dolphins. There are 9 dolphins in this slide. Can anybody see a dolphin? Oh my God, what an audience. There should be dolphins here. But can anybody see a dolphin, please? See the nose here? There's a dolphin. Oh, don't dwell on it, maybe. Okay, anyway, one of the things that I love about coming to India and other countries is that we learn from one another. Through UNESCO, we've been able to run some international courses. Here's one in Petra. At Woods Hole, every summer, every fall, we have a course on reproduction and another course on stem cells and regeneration. And I know we can't accommodate all of you, but if any of you are interested, please let me know and we'll get you into the application process. And thank you very much for your attention. I'd especially like to thank Cal Simmerly, Jadranka Lonkarik, and Tomar Vidor-Reis, as well as Pasquale Patrizio. Thank you very much.